Welcome back, Life Science learners, to our final installment. We've looked at the process of DNA replication. We've tried to unpack that process by looking at the different processes that occur to produce what we refer to as two identical copies. This process is a highly accurate process. There's significant proofreading of the DNA, which ensures that any mistakes that do occur when that template is being copied is corrected. We do know that these mutations do happen, and they often occur in more smaller organisms that have a really small genome that reproduce exponentially. So if we were looking at mistakes that do happen in the process of DNA replication, we refer to those as mutations. We're not focusing on mutations today, but it's important that we recognize that mutations do occur. But the rate at which they occur often takes us to the next process of trying to understand why is it that mutations do not occur as often as it would in the process of copying DNA. And so that takes us to the next process of looking at the models that explain DNA replication. And because of these models, we have a better understanding of why DNA replication is such an accurate process. So the first model that we're looking at is called the semi-conservative model. The second model is called the conservative model, and the third model is a dispersive model. And these models essentially try to explain the process of how replication can be looked at. An overview of these can be illustrated using simple colors. So right on the top here we have the semi-conservative model, and it produces two models of DNA as you see here. What is unique about these models is that if you look at this or these two new strands that are produced, each one of them has part of the original, which is in yellow, and part of a new, which is in blue. So we refer to this as a model showing half, which is semi, and conservative is referring to a term where we refer to keeping the old. So at the end of this process of replication using the semi-conservative model, we have two new strands, and each of these strands have a copy of the old strand and a new complementary strand. And so this is referred to as the semi-conservative model. The second model that we look at is the conservative model. And if you think of the word conservative, it essentially refers to keeping the old. And so the conservative model talks about DNA being replicated, where the original model is intact and a new copy of DNA is produced. And this strand is an entire new strand that is made up of entirely new produced or synthesized nucleotides put together into a form of DNA. And so scientists have rejected this purely based on that because of the frequency at which mutations occur, that this possibly is a model that will create a lot of faults in the DNA, essentially lots of mutations. A third model was also proposed, which was called the dispersive model. And this model talks about the new structures of DNA or strands of DNA having segments of old and segments of new dispersed within them. And so this model is found also to be erroneous, meaning prone to mistakes, and hence these two are the models that are not essentially accepted, whereas this model, the semi-conservative, is the one that is recognized as the most consistent model to explaining the accuracy of the process of DNA replication. So we refer to that as the semi-conservative, which produces new strands, each containing one of the original strand and one new strand. And so we're going to move on to trying to apply our understanding to this in terms of some questions. So let's get straight into these questions. The diagram represents a part of a molecule. Study the diagram and answer the questions that follow. So I'm going to call that diagram up in a bit. So here we've got a diagram of a DNA molecule that's illustrated. Let's try and understand firstly why this is DNA, and I can see that it's double-stranded. I also can see that there's possibly complementary base pairs, which I know that we will have to unpack in a little while. So that's an overview of that diagram. There's a key that's been provided, and that key again refers to the specific shapes and names of these nucleotides. So that's what's been given to us. Let's look at the questions based on this diagram. So the first question, A, identify the molecule in the diagram above. And so when I look at that molecule there, remember it's referring to the type of nucleic acid, and so that is DNA. And simply I can say that because I can see that it is a double-stranded structure. More important than that, we know that DNA would contain a thymine base, and we're going to try and find out if there's a thymine base present on that. The next question is label the parts numbered 1 and 5 respectively. So guys, it's important that you look at the question mark allocation. It requires that we label parts 1 and 5. It does not say 1 to 5. And so if you were labeling parts 1 to 5, it would mean that you would have spent an extra few minutes on questions that were not necessary. So it's important that you recognize that it requires that we label parts 1 and 5. So let's get back to this image. So we've got to label parts 1, and I'm going to identify quickly part 5. And so this would require us to be able to access the key. If we go to the key, we know that A represents adenine. So it means that if we look at adenine, which is clearly indicated at this position, it would mean that number 4 would be thymine. Okay? Using that, we know that again, that these bases would be, wherever you see that, there's going to be, let me get this right, a thymine there, and that's going to be an adenine based on its shape being similar to this base here. And we would know that if you've got guanine here, you've got to have cytosine complementary to it. And likewise, based on its shape and the similarity between this and number 5, we would know, and I can infer that number 5 has to be a cytosine base. So we look at number 1. Number 1 points to specifically the structures between A and T, and those would be the hydrogen bonds. So number 1 would point to the hydrogen bonds, and remember that these nitrogenous bases are held together by the hydrogen bonds. Okay, so that's what we're looking at for number 1. The next structure that we needed to identify was number 5, and I think it's specifically pointing to the skill of you being able to identify the type of nitrogenous base. Yes, number 5 is a nitrogenous base, but I think the question wants us to be very specific about the type of nitrogenous base, and using our inference that guanine and cytosine base pair, and looking at that shape, we can confer that number 5 refers to cytosine as the nitrogenous base. Cool. Let's move on to a few more questions. What is the collective name for the parts numbered 2, 3, and 4? So if we look at parts 2, 3, and 4, again, so I'm going to go to 2, 3, and 4. So we're going to put 2, 3, and 4 together. So 4 would be here. If we looked at them together, we know that a DNA nucleotide consists of 3 parts. It consists of a sugar, it consists of a phosphate molecule, as well as a nitrogenous base. So number 2 is the phosphate, number 3 is the sugar, and number 4 is the nitrogenous base. And so these 3 parts collectively are referred to as a DNA nucleotide. And we refer to that as a monomer of DNA, or a building block. Cool. The next question. What is the significance of this molecule being able to replicate itself? Now, the question refers to what is significant about DNA being able to replicate itself. And so if we understand the model of replication, and the model that we referred to was the semi-conservative model of replication, we can understand that simply that the structure of DNA with its complementary nature allows for free nucleotides to easily base pair when the strands are separated. So based on the unwinding and the separation into the templates, we know that these templates now form an easy access point for the free floating DNA building blocks to now base pair with their complementary nature. And so this talks to the structure of DNA, allowing for the ease at which it can copy. So the significance of this molecule being able to replicate is that DNA is double-stranded. And so when the strands separate, each of these strands forms a template, allowing for complementary bases to base pair with these templates. And so it's important that we recognize that due to the double-stranded structure as well as the complementary base pairs, that the process of DNA replication using the semi-conservative model of replication becomes efficient and a reliable method or model of replicating DNA. Right, so we'll move on to the next question. The diagram represents a portion of a nucleic acid. Okay, so here's a diagram that represents a portion of a nucleic acid. I'm going to try and read more into the question. Name the nucleic acid for 1 mark, and the second question is name 2 places in animal cells where this nucleic acid may be found. So let's try and identify which nucleic acid is illustrated in this image. If we look at that image closely, we can see that it's double-stranded. Again, that double-stranded nature of DNA is a distinguishing characteristic of DNA being double-stranded, and hence I use that as a significant point to be able to identify that DNA is the nucleic acid. Cool, that's a 1 mark question. Name 2 places in animal cells where this nucleic acid may be found. It's important that we recognize that firstly, we have DNA in the nucleus, so we refer to that as nuclear DNA or chromosomal DNA, so it can be found as chromosomal DNA or in the nucleus. But also we know that DNA can also be found in the mitochondrion. And if we look at the mitochondrion, obviously that's the cell, that's the organelle that controls the process of cellular respiration. And so DNA is found in there, and it's linked to providing the instructions to be able to produce important enzymes and controlling how the mitochondria function. The next part of the question requires us to identify different parts in this. So question C, identify portion V, W, Y. Okay, so let me read that again. We've got to identify portion V. We've then got to identify the nitrogenous base W, the molecule Y, and the bond Z. So a few things that we've read together. We're going to go back to this image and we're going to unpack each one of them. So if we look at V, and V again collectively points to all of these 3 structures together. And as I mentioned, if we look at these 3 structures, it's the 3 components that make up the building block of DNA. So V would be a DNA nucleotide. Okay? The next part that we need to identify is the bond Z. And as I mentioned that these bonds which are the hydrogen bonds, hold the base pairs together. So that's your hydrogen bond. Okay? Y would be pointing to the phosphate molecule. So we know that DNA contains a sugar phosphate backbone. Okay? X would be pointing to the deoxyribose sugar, and it's fine for us to indicate that as a sugar, but it's actually pointing to this type of sugar, which is a deoxyribose sugar. And W in this case would be specific to what is being given on the 1 template. So that's a cytosine base, which would refer to this being a guanine nitrogenous base. And that is essentially the structures that we needed to label. The next question, what is the natural shape of this molecule? And due to the chemical bonding between the complementary base pairs, DNA takes on a helical shape. And so the natural structure of DNA is that it is helical. We move on to this last question. Name the process during which this molecule makes a copy of itself. And guys, we've spent the second half of this lesson today looking at the process of DNA replication. And so DNA replication is that process where DNA is copied to produce identical copies of itself, and we refer to that model as the semi-conservative model of replication. So guys, as we wrap up this section, a quick overview of today's lesson. We've looked at basically the structure of DNA as an overview. We've looked at the process of DNA replication. We've also identified the role of DNA in terms of as an important coding protein or coding nucleic acid in the cell. It's important that we understand that DNA replication takes place, when it takes place, and where. As a recap, DNA replication requires a complex series of enzymes that carry out this process to produce exact copies at the end. And this all happens so that the cell can prepare for cell division. So guys, you've done well, you've been focused. I hope that you've understood the process of replication. I encourage you to go back, read some of your notes, and try and make some of your own questions and answer them. So all the best, go well, keep safe.